DNA fingerprinting. As started in the preceding section, 99.9% of the base sequence among the human is the same, assuming human genome as 3 into 10 raised to power 9 base pair. In how many base sequence would there be difference? In it is the difference is sequence of DNA, which make every individual unique in their phenotypic appearance. If one aims to find out genetic difference between two individuals or among individuals of a population, sequencing the DNA every time would be daunting and expensive task. Imagine trying to compare two sets of 3 into the power of 6 base pair. DNA fingerprinting is a very quick way to compare DNA sequence of many two individuals. DNA fingerprinting involved identifying differences in some specific region in DNA sequence called as repetitive DNA, because in this sequence is small stretch of DNA is repeated many times. This repetitive DNA are separated from the bulk genomic DNA as different peaks during density gradient configuration. The bulk DNA form a major peak. The other small peak are referred to as satellite DNA. Depending on the base composition, A raised to T rich or G raised to C rich. Length of segments. The number of repetitive units, the satellite DNA is classified into many categories, such as microsatellites, mini-satellites, etc. The sequence normally do not code for any proteins, but they form a large portion of human genome. The sequence show high degree of polymorphism and the basis of DNA fingerprinting. Since DNA from every tissue such as blood, hair, follicle, skin, bone, saliva, sperm, etc. form as individual show the same degree of polymorphism, they become very useful identification tool in forensic application. Further, as the polymorphism are inheritable from parents to children, DNA fingerprinting is the basis of the paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetics mapping of human genome as well as DNA fingerprinting, it is essential that understand what DNA polymorphism means in simple terms. Polymorphism verification at genetic level arise due to the mutation. Recall different kind of mutation that the effect that you have already studied in chapter 4 and in preceding section. In this chapter, new mutation may arise in an individual, either an somatic cell or in the germ cell that generate gametes in sexually reproducing organism. It is a germ cell mutation does not seriously impair individuals or ability to have offspring who can transmit the mutation. It can spread to the other membrane of population through sexual reproduction. Allelic, again recall the definition of allele from chapter 4. Sequence variation has traditionally been described as a DNA polymorphism. If more than one variant allele at a locus occur in human population with a frequency greater than 0.01, in simple terms, if an inheritable mutation is observed in a population at a different frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non-coding DNA sequence will be higher as mutation in the sequence may not have any immediate impact in an individual's reproductive ability. Thus, mutation keeps an accumulating generation after generation and from one of the basis of variability or polymorphism. Thus is a variety of different type of polymorphism, regaining from single nucleotide change to very large scale changes. For evaluation and speciation, such polymorphism play a very important role and you will study this detail at higher classes. The technique of DNA fingerprinting was initially developed by Alex Jeffrey. He used as a satellite DNA as a probe and show very high degree of polymorphism. It was called as variable number of tandem repeats VNTR. The technique as used earlier involves southern blot hybridization using radio labeled VNTR as a probe. It include first isolation of DNA, second digestion of DNA by restriction into nucleus, third separation of DNA fragments by electrophoresis, fourth transferring of separated DNA fragment to synthetic membranes such as nitrocellulosic or nylon, fifth hybridization using label VNTR probe. And 6. Detection of hybridized DNA fragment by autoradiography. A schematic representation of DNA fingerprinting as shown in figure 5.16. The VNTR belong to a class of satellite DNA referred to as mini satellite. A small DNA sequence is arranged randomly in a many copy number. The copy number varies from a chromosome to chromosome. In an individual, the number of repeats show very high degree of polymorphism. As a result, the size of VNTR varies in size from 0.1 to 20 kb. Consequently, after hybridization with VNTR probe, the autoradiogram give many bands of binding sizes. Thus, bands give a characteristic pattern for an individual. DNA figure 5.16. It refers from individual to individual in a population, except in the case of monozygotic, that was identical twins. The sensitivity of the technique has been increased by use of polymerase chain reaction (PCR). You will study about in chapter 9. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. In addition to application of in forensic science, it has much wider applications such as in determining population, the genetic diversity. Currently, many different probes are used to generate DNA fingerprints. Thank you. The topic was end.